Hi everyone, it's Ms. Lopez. In this video, we are going to continue with chapter 2 and 3 with the macromolecules. This time we're going to be looking at the nucleic acids. So some key things that we're going to be looking at for these nucleic acids include the structure of the nucleic acid. We're going to talk about how the nucleic acids are formed. And then in the next part, in the next video, we will focus on proteins. So nucleic acids are polymers specialized for storage and transmission and use in genetic information. DNA and RNA are the two most common examples of those nucleic acids, although ATP, which is energy and has stored chemical energy in its bond, is also considered a nucleic acid. DNA stands for deoxyribonucleic acid. You can always just use the abbreviation DNA without having to write out the whole word. And RNA is ribonucleic acid. And again, you can always just write RNA without having the write out the entire word. And then just a reminder that the nucleotide or the monomers for nucleic acids are nucleotides. So a nucleotide is made up of three things. Number one, a sugar, and it's going to be a pentose sugar, meaning 5 carbons. It's also going to have a nitrogen-containing base and a phosphate group. So the three things that make up the nucleotide are the pentose, the sugar, the nitrogen base, and the phosphate group. You would also have what are known as nucleosides, and you can see that they also contain the sugar and the nitrogen base. They're just missing that phosphate group. And so down here, they're showing you how it gets built. You've got your base, plus the sugar is the nucleotide. Adding that phosphate group will give you a nucleotide, which is your monomer for nucleic acids. The bases for the nucleic acids are divided up into either pyrimidines or your purines. So your pyrimidines are single rings, the purines are double rings, and the sugar can either be deoxyribose or ribose. So I'll go into detail about the bases a little bit further down, maybe even on the next slide, but I want to talk to you guys about the sugar because it's a great example of how structure affects function, which is one of our big themes throughout all of AP biology, is the difference in this sugar. So it's a pentose, meaning we have our oxygen and 5 carbons. So this here is carbon number 1, number 2, number 3, number 4, and then here's carbon number 5. So the difference between deoxyribose and ribose is the presence of a hydroxyl group. And that hydroxyl group is specifically at carbon number 2. So if it contains the sugar, if this sugar contains that hydroxyl group at carbon number 2, then this is considered ribose. If that hydroxyl group at carbon number 2 has been removed, then this is considered deoxyribose. So deoxyribose and ribose have everything else the same. You know, the difference between them is the presence of a hydroxyl group at carbon number 2, and that hydroxyl group would create instability in the molecule. As a result of that instability, RNA is going to be less stable. And because it's that oxygen, that hydroxyl group is missing in DNA, in that deoxyribose, DNA is much, much more stable. So another great example of the difference of how the structure affects function for these molecules. This is a graph that's showing the different types of nitrogenous bases that can be found in the nucleotide. So in that nucleotide, the nitrogenous base is going to be attached here to carbon number 1. Here's that oxygen, here's carbon number 2 we just talked about, here's carbon number 3, number 4, and then there's number 5. And that nitrogenous base is categorized as either a pyrimidine or a purine. So you don't have to memorize the structure of the pyrimidine versus the purine, but you do need to be able to identify which ones are the pyrimidines and which ones are the purines. So your pyrimidines are cytosine, thymine, and uracil. And so one way that I try to help students remember that which ones the pyrimidines are, TCU, like the college in Texas. The other two nitrogenous bases are the purines, and these ones are adenine and guanine. So AG, ag, like the Aggies. So it's the Aggies versus TCU. So your pyrimidines have one ring. And when I say one ring, watch. I'm talking about this carbon, this structure that has this shape. So we have a carbon here, and it goes like this. It's not in a long linear form. It's what's called a ringed structure, because it forms kind of like a ring. So pyrimidines have one ring, and you'll see here that your purines have two rings. There's one right here and one right here. Same thing with guanine over here. One ring, two rings, so it's got two rings and it's considered a purine. Nucleotides bond in condensation reactions to form those phosphodiester linkages. Remember, condensation reactions, also known as dehydration synthesis, remove water. And those nucleotides are going to grow in what we call a 5-to-3 direction. So we're not going to get into too much detail at this point, but we certainly are in January when we go into DNA and the structure of DNA and why it's important to denote the difference between the 5 and the 3. So in 9th grade, whenever you guys would do DNA matching or DNA growing, you would write 3 to 5. And you would write a sequence, let's say A, G, T, C, A, okay, just randomly. And then if you had to come in and put the anti-parallel or the complementary strand, you would put 5' to 3', and then you would match them. So A pairs with A pairs with T, G pairs with C, T pairs with A, C pairs with G, and then A pairs with T. So you would match them up, and then you would be, good to go. What those numbers are referring to is the carbons that are on the deoxyribose or the ribose. So if we draw one more time, the deoxyribose, this is carbon number 1, carbon number 2, 3, 4, and this is carbon number 5. Again, I know this is deoxyribose because right here at carbon number 2, I didn't draw a hydroxyl group. That's how I know that this is deoxyribose. So this 3 to 5 is referring to where we are bonding the next nucleotide. And so we always, we're always going to add it to that 3' carbon. So carbon number 1, 2, 3. When we make that phosphodiester bond, we're going to add it and remove the water from this position right here. Again, we'll talk about more details in that in January. For now, just know that those numbers are referring to the carbons that are in this structure. Here are some things that kind of help you to distinguish. This is a great FRQ question where I ask what's the difference between RNA and DNA. So in RNA, the sugar is ribose, and the bases are A, C, G, and U for uracil. And RNA is always going to be single-stranded. In DNA, the sugar is deoxyribose, and we have adenine, cytosine, guanine, and instead of uracil, we have thymine, and DNA is double-stranded. Your complementary base pairing is between the nitrogenous bases themselves. So between the nitrogenous bases, you're going to get adenine and thymine that pair. And what I want you to take note of here is that you notice that there's some hydrogen bonds, they're labeled hydrogen bonds, here between these two. And so thymine and adenine will form 2 hydrogen bonds between them. Cytosine and guanine have 3. So see the picture down here? It's got 3 lines to show you that there are 3 hydrogen bonds. The more hydrogen bonds there are, the more connected with the 2 molecules are going to be. And so when we look at the shape of the DNA, those hydrogen bonds are going to form attraction and hold the strands together. So because there are so many, DNA can be something like 10 to the 18th base pairs. That's a lot. And in between them, there are multiple hydrogen bonds. So collectively, hydrogen bonds are considered fairly strong, although individually they are weak. So with DNA, it's not going to take that much energy to break that bond because they're not true bonds. They're just hydrogen bonds. They're just attraction between the nitrogenous bases. And so you can easily separate DNA. And when I say easily, I say compared to like breaking the bonds in carbohydrates or breaking the bonds in proteins. And so the amount of energy that's going to take to separate out the DNA strands when it comes time to replicate it is much less than it would be than to say break a molecule. RNA strands are usually single-stranded, but they can be folded into 3D structures based on hydrogen bonding. It can kind of fold in on itself. And so because of the different order, whether you write we're not going to write T-A-U-G-C, or if you write C-C-A-G, that order in which the nucleic acids are laid down is going to influence how that shape and how that folding happens. DNA is considered that double ladder, that twisted helix, and those sugar phosphate groups form the backbone. So here is my sugar phosphate group, and then those nitrogenous bases would be in between, sugar, phosphate, and then in here, in the middle of the ladder, right here, what we call the rungs of the ladder, this is where those nitrogenous bases would be. And if it's A and T, they'll form 2 bonds. If it's C and G, they'll form 3 bonds in between. So this part right here is the same for every organism on Earth. The sugar phosphate, everything. What's different and what makes you different is the order in those nitrogenous bases, the specific sequence of those nitrogenous bases. So this is kind of a picture hopefully you guys are familiar with. This is the anti-parallel strands where you have your C and your G, your A and T together of the DNA molecule. We are going to go into a lot more details about the structure of DNA, but not until January. For now, your knowledge of nucleic acids as a macromolecules is sufficient for Unit 1. Thank you guys so much for watching. I hope this video was helpful, and we'll see you guys in the next one. Bye.